Okay, imagine this. You're a scientist, and your job is to find the toughest, greenest grass in the world. You're staring at literally thousands of little green squares of turf, and you have to pick the winner. Sounds kind of simple, right? Well, it turns out it's a huge challenge in agricultural science, and it pushes researchers to get really, really clever about how they get their answers. So let's dive into this quest for perfect data.
So take a look at this. This is the view from above. Every single one of those squares is a different type of grass, or it's getting a different treatment. Maybe more water, maybe less. For years, the way this worked is a researcher would literally walk these fields and just rate them by eye. This one looks like a 7. That one's maybe an 8. But our eyes are tricky. They can be fooled by the time of day, by a passing cloud, even by how we're feeling. It's totally subjective. So the real question is, how do you turn that feeling into hard, objective data?
Well, the answer lies in technology, in moving from pixels to plots. In this first part, we're going to see how researchers took the human eye out of the equation and brought in a much more precise digital one to turn simple pictures into some really powerful data.
The solution is actually pretty brilliant, and it's something most of us have in our pocket, a regular digital camera. By just taking a picture of each plot, researchers can feed those images into a computer, and that's where the magic happens. The software does all the heavy lifting, analyzing every single pixel to objectively measure things like how much of that square is actually green grass versus just brown soil, and even the exact shade of that green.
And one of the key numbers they get from all this is something called the DGCI, which stands for Dark Green Color Index. It's pretty much exactly what it sounds like. It's a score for how green the grass is. So a higher DGCI number means you've got a darker, richer, and usually a much healthier plant. This right here is our first piece of solid, unbiased data, pulled straight from the pixels.
And you know what? Right off the bat, this method totally proves its worth. If you look at this chart, you can see the DGCI scores for plots that got different amounts of water. The ones labeled IRR 100, they got all the water they needed. IRR 50 only got half. And just like you'd guess, the happy, well-watered plots have a much higher greenness score. This is a really important point. Even without any fancy corrections, this simple photo analysis is already working. It can tell the difference.
So, the basic method works. We've established that. But scientists are never happy with just good enough. They're always chasing perfect. And that brings us to the real heart of this whole thing, a test of true color. Could they make this already good data even better?
So here's the problem. Here's the catch. A digital camera, just like our eyes, can get fooled by light. Think about a photo you take at noon on a bright sunny day versus one you take later on a cloudy afternoon. The colors look totally different, right? Well, the same thing happens here. So the big question for the researchers was, what if we could correct for those lighting changes? Could we make our data even more precise, even more reliable?
And this is the tool for that exact job. It's called a color checker card. Honestly, the best way to think about it is like a ruler, but instead of measuring inches, it measures color. Each one of those little colored squares has a scientifically perfect, exact value. So by sticking this card in the photo, you give the computer a perfect reference point to work from.
And you can see what the software does right here. It looks at the color checker in the photo, it compares the colors it sees to the true values it already knows, and then it adjusts the entire image to match. See that? Look at the corrected image. The software tweaks everything until those little squares are perfect. You can notice how some of the colors, especially that blue one, get a little deeper, a little darker as they're pulled into perfect alignment.
Okay, so the theory is solid, and the correction works perfectly on the little card. But what happens when we apply it to the actual grass, to the turf plots? Well, now it's time to follow the data and see what really changed when we compare the original photos to the corrected ones. All right, here we've got a turf plot before and after the color correction. Just take a second. Can you spot the difference? It is incredibly subtle. I mean, to our eyes, it's pretty much impossible to tell these two pictures apart. But what about the computer's eye?
This is what the computer sees. It has gone in and isolated every single pixel that it thinks is green turf. And again, we're looking at the original versus the corrected. Even for the software, the difference in the amount of green it finds is tiny. In this particular plot, which was actually one of the ones with the biggest change in the whole study, the measured green cover actually went down by about 1.9% after the correction.
And that wasn't just a one-off. When they averaged it all out, across all 912 plots in the study, color correction didn't increase the measured turf cover, it actually decreased it, on average, by 0.5%, a really small but consistently negative change.
This table breaks it down even more, and honestly, it shows a really mixed bag. Turf cover and our greenness score both went down a little bit, but then saturation and brightness went up a tiny bit, and the amount of blue the camera detected dropped quite a bit. Basically, the correction made the image a little less green, but maybe a bit more vibrant and less blue. The point is, there's no clear, consistent improvement here. The changes are small and they're pulling in totally different directions.
Now here's the real test. The researchers compared their photo data to something called NDVI, which is a standard way to measure plant health from satellites. The idea is if the correction is making the data better, it should line up more closely with the satellite data, right? Well, as you can read here, the exact opposite happened. The correlation actually went down a little bit. That's a huge clue that this extra step, well, it didn't really improve the quality of the data in any meaningful way.
So where does all this lead us? It leads us to a really surprising, and I think a really useful verdict. And it begs the question, is better always actually better?
Let's just look at that original uncorrected data one more time. This chart is plotting the satellite data against the simple photo greenness score. See that R-squared value of 0.55? All that really tells you is how well those two things line up. A 1 would be a perfect match, so 0.55, that's actually pretty good. It shows there's a real solid relationship there. The simple, uncorrected method really does work.
So, the big finale. Did that extra, complicated step of color correction make the data any better? The researchers checked, and the improvement in the correlation was 0.001%. Nope, that is not a typo. For all practical purposes, the impact was 0. All that extra processing for a statistically invisible change.
So what's the final verdict? Well, first, it's crystal clear that color correction had a minimal, basically negligible impact on the results. Second, the changes it did make were tiny and all over the place. But most importantly, this whole thing proves just how strong and effective the original, simple method really is. It's a fantastic reminder that more processing, more steps, and more complexity doesn't automatically mean you get better data.
And that leaves us with a much bigger question, one that goes way beyond turfgrass. In science, in our jobs, in life, we're always chasing perfection, always looking for more accuracy. But this study is just a great reminder to stop and ask ourselves, in that endless search for perfect data, how do we know when good is already good enough?